Let's look at where the receptors for the hormones are located and what happens once the hormone binds to its receptor. We're going to begin with steroid-based hormones. So let's look at the steroid-soluble hormone actions. Because these hormones are nonpolar and lipophilic, they can go right through the phospholipid bilayer of the plasma membrane. This means that the receptor for the hormone is located within the cell, either in the cytoplasm or in the nucleus, such as in this illustration. When the hormone binds to its receptor, it forms a hormone receptor complex. The hormone receptor complex next binds to the DNA to activate a gene or genes. This brings about protein synthesis. Either a polypeptide or a protein will be synthesized in response to a steroid hormone. Water-soluble hormones behave differently because they are hydrophilic. Since they're hydrophilic, their receptors have to be located on the exterior surface of the target cell membrane. The binding of the hormone to the receptor can lead to three possible outcomes. Let's take a look at these outcomes. The first outcome involves a G protein and the formation of a second messenger. The first messenger, these are chemical messengers, is the hormone. It doesn't enter the cell, it just binds to the receptor on the external surface. But this leads to the formation of a second chemical messenger located within the cell. It's the second chemical messenger that is going to bring about the cell's response. In other words, it's this messenger that's going to trigger a series of reactions leading to the end product or products. The binding of the hormone to the receptor causes the protein receptor to change its structure. Recall that structure determines function. The structural change exposes a binding site for another protein called a G protein. The G protein has three subunits, an alpha with a guanosine diphosphate bound to it, a beta subunit, and a gamma subunit. The structural change that occurs in the receptor is used to relay a message to the interior. This is signal transduction. When the G protein binds to the receptor, the GDP is replaced by guanosine triphosphate. This leads to the separation of the alpha subunit from the other two subunits. The alpha subunit will bind to an inactive form of an enzyme called adenylate cyclase. Notice all of this is happening along the plasma membrane. Adenylate cyclase removes two phosphates from ATP, converting it into cyclic AMP, and cyclic AMP is your second messenger. What occurs next is dependent upon the enzymes present in the cell. Phosphodiesterase will inactivate cyclic AMP. If, however, the cell has an inactive form of an enzyme called a protein kinase, cyclic AMP will activate the protein kinase. A protein kinase is an enzyme that phosphorylates other proteins. The phosphorylation of other proteins will activate those proteins to function as enzymes. And an enzymatic cascade begins, similar to coagulation. This leads to the cell's response. Hormones, therefore, can have different end results depending upon the cell type and the chemicals located within that cell. Signal amplification occurs when the second messenger phosphorylates multiple protein kinases, which are your secondary effectors. And each protein kinase, in turn, phosphorylates multiple proteins. So now you're going to have many enzymatic cascades occurring, producing hundreds to millions of molecules. You have multiplied your effect or amplified it. The second possible outcome also involves a G protein, but in this case, the G protein will be used to either open or close a membrane channel. The alpha subunit will bind to the channel. Recall that channels are proteins as well, causing it to change its shape, either into the open position or the closed position. Recall, each channel is specific as to the type of substance that can go through it. What we're looking at here in this illustration is a calcium-gated channel. The alpha subunit has opened up that calcium-gated channel, so calcium now can move in. We can have an influx of calcium. The third type of outcome does not involve a G protein. In this case, the receptor, once it's activated, becomes an enzyme. In this illustration here, you can see the binding of the hormone to the receptor will cause it to change its shape, exposing binding sites for phosphates. When the phosphates bind to the receptor, it activates the receptor, and now it functions as an enzyme to phosphorylate other proteins that are located within the cell. And these phosphorylated proteins then will bring about the cell's response. So it's a direct response because it does not involve a G protein. The receptor functions as an enzyme itself. Target cells regulate their response to a hormone or their ability to bind with a hormone in one of two ways. They can increase the number of receptors that they have. By increasing the number of receptors, the cell is going to increase its responsiveness to the hormone. This is called upregulation. A cell may do this if blood hormone levels are low. Downregulation is just the opposite. In this case, a target cell can decrease its responsiveness to a hormone by decreasing the number of receptors. So if you have elevated levels of a given hormone in the blood, you might want to decrease the number of receptors, thereby decreasing the target cell's response.
There are three types of hormone interactions: permissive, synergistic, and antagonistic. A permissive interaction occurs when one hormone requires the presence of a second hormone to exert its full effect. For example, thyroid hormone has a permissive effect on the actions of reproductive hormones. Thus, a lack of thyroid hormone can delay reproductive development. During synergistic interactions, multiple hormones bring about the same actions. However, their combined effects are amplified. For example, the hormones glucagon and epinephrine both increase blood glucose levels. When they act together, the amount of glucose is increased 150% of what occurs when the hormones act alone. An antagonistic interaction involves two hormones with opposite actions, such as insulin, which decreases blood glucose, and glucagon, which increases blood glucose.